Okay, my name is Arden and this is my current event article titled CRISPR-Cas9 Effectors Facilitate Generation of Single-Sex Litters and Sex-Specific Phenotypes. So a little bit of background information. For the article, the researchers use CRISPR-Cas9 technology, which, as we discussed in class, is able to cut and edit DNA of organisms. And part of the researchers introduced a gene lethality for each sex. So basically, if the genes were going to code for the males, sometimes they would introduce gene lethality for the males, so that the embryo wouldn't develop and would just kind of die off, or the opposite for the females. And with this technology, and the researchers were able to produce female or male only offspring with 100% efficiency. And after they used the CRISPR technology, they did a PCR test and different types of genotyping to examine the genomes of the mice. So who and where? The who and where is the researchers at the Francis Crick Institute. And there was a handful of researchers, and I included a couple right there. And the type of organism that they were testing on were mice, specifically newly fertilized mice embryo, where they were using the sex chromosomes of the mice and the CRISPR technology to alter the chromosomes and introduce some gene lethality. And who was affected by this research? Farmers specifically, and researchers, scientific researchers. So, which I will talk about more in the next couple of slides. And when was this article produced? It was published December 3rd, 2021. So it is a fairly recent article with fairly recent research. So some major findings that the researchers had is that they were able to use CRISPR to control the sex of offspring and litters from the mice. Specifically, they were targeting the sex chromosomes, and they introduced different types of gene lethality. So they would affect either the X chromosomes or the Y chromosomes of the male or female to introduce the lethality to where the embryo would no longer develop. And we're able to do this at 100% efficiency. And the offspring that they had when they introduced the certain genes were either 100% female or 100% male. And the surviving offspring had no effects because of the CRISPR gene editing. They were fully functional, and it basically couldn't tell that they had anything done to them at all. And one of the major findings was how easy and applicable this could be to other types of species. And the way that they found this to be applicable was with the top1 gene. So the top1 gene is a gene that is widely found in other types of genomes for other species, and could be definitely proposed with more research for them. And why is this research relevant? Well, the first would be for scientific researchers, and how safe and effective this was. They had no issues with the offspring, and they had 100% efficiency. So there was never a mistake where they were trying to control for only female offspring and they happen to have a male. Never happened in their research, and they were always successful. And could be used for the researchers for multiple reasons, but including if they were studying reproduction and females of a certain species and they wanted to only use females in their research, and the offspring, they could use this technology to where they'd only have female litters. So they'd always would have females to use. They wouldn't have to do anything with the males. Or if they were studying sex-specific diseases in males, perhaps other females, they would always be able to control the litter, the sex of the litter of the offspring. And for farmers purposes, certain male or female sexes have specific purposes on the farm, whether that is in dairy production in cows or egg production and chickens, for example. It's like if the farmer only wanted to have chickens, so they could produce lots of eggs, they could control the litter to be 100% females, where they wouldn't have to do anything with the males. And in The article, it talked about how culling is a major issue with scientific research and farming, where all the leftover offspring just end up being killed off or gotten rid of. And ethically, this produces problems with having to just kill innocent animals that weren't a desired sex. So this research would pertain to that. Future research, there wasn't a lot about future research, but what they did say is that this could be used with other species. Kind of with that top one gene, is that if this works for mice, what other types of species would this be able to work for? So more research to test whether or not this would be something that could be used for other types of animals or even humans possibly. And also developing commercial methods. If farmers and researchers want to use this, the CRISPR technology and is not cheap for everyone to be able to just use. So they would need to develop methods and where it would be very accessible for everyone to use and affordable. And these are my references.